Okay, so in this video I'm going to cover Chapter 4, Cell Biology, cell structure. So we're going to be going over the parts and pieces of cells. So first off, you know by now that cells are the basic unit of life, right? We talked about the hierarchy of living things. So we said that kind of at the base of that we have atoms and molecules make up organelles, and then organelles make up cells, right? So that's where we are right now. Atoms and molecules make up macromolecules. Macromolecules make up organelles, and organelles make up cells. So we're going to talk about things at the cellular level now, and we'll be talking about the organelles. And then you know that cells, if we're talking about a multicellular organism, cells are going to make up tissues, and tissues make up organs, and organs make up organ systems, which make up organisms. Okay? Now, that's a key thing I said there. In multicellular organisms, we have tissues, but in unicellular organisms, we do not. So let's first go over those differences. So again, cells are the basic unit of life. They're going to be the smallest thing that we consider to be living, which means that if we take out the parts and pieces, the organelles, or the molecules themselves that make up organelles, those things are not considered to be living. Cells are. So they are the smallest unit which is considered to be alive. So they are the basic unit of life. Cells come in two types. Okay, so we have prokaryotic organisms on the planet, and so prokaryotic cells, and then we have eukaryotic organisms, which have eukaryotic cells. So that we're going to talk about the main differences between prokaryotic cells and eukaryotic cells. And organisms can be either single-celled or multicellular. So prokaryotes are going to be single-celled organisms, and many eukaryotes are multicellular, but not all. There are some single-celled eukaryotic organisms. We have to be careful there with that definition. This figure is straight out of your book. It's showing you some, mostly, multicellular organisms, and then we've got a figure there showing some single-celled organisms. And so if we look here, let me grab my pointer. So here in this first figure, again, we're showing multicellular organisms. I forget exactly what tissue this is taken from, but it's taken from a tissue of a multicellular eukaryotic organism. Okay, an organism made up of many cells, and whose cells have membrane-bound organelles, which we're going to get into in this chapter. This second figure is from a plant. You can see that these cells are rectangular in shape, so let me just kind of outline a cell for us here. That's one cell. So there's multiple cells on this figure. That one cell has a very rigid rectangular shape in this case, definitely indicating that there's a cell wall there. And you can see this little circular structure here. That's the nucleus, right? That membrane-bound compartment, which we're going to learn about in this chapter. And then here in this last figure, these look like single-celled bacterial cells. Okay, prokaryotic. Okay, so again, we're going to learn the differences between prokaryotic and eukaryotic cells and look at the components of cells. So before we get into that, I need to introduce you to microscopy. So cells, whether they're from a prokaryotic organism like a bacteria or a eukaryotic organism like us, when we try to look at our cells, we can't see them with the unaided eye. Instead, we need an instrument like a microscope. And unfortunately, you haven't been able to actually use a microscope. But in lab week 2, we introduced you to the microscope. And so you at least got to see it. And you know that microscopes are going to be used to magnify an object, to make something appear larger. Okay, so again, most cells are microscopic, which means you cannot see them with the unaided eye. A microscope, which is shown here in this picture, is going to be an instrument that's used to make an object appear larger or magnify that object. And some microscopes will have a camera on them, which Again, if we were in the lab, my microscope, which I miss, has a camera on it, a beautiful camera on it, where I can actually take whatever I'm looking at under the microscope and I can project it onto a screen in the front of the classroom, which is really neat so that you can see what I see. And sometimes you can even take a picture. And a picture taken with a microscope is called a micrograph. Okay, so for example, on the previous slide, these were micrographs. These are images taken with a microscope. It's pretty neat. Now, the light microscope, which is the microscope that's used in most general biology classrooms or even in high school biology classrooms, we use a light microscope, a compound light microscope. So what does that mean? So if we take a look here in the figure, this is a compound light microscope. Here's where you would look. So there's your eyeball. And the microscopes that we have in the lab are binocular. So we actually have two. You can look through two. It's called the eyepiece. Okay, it's a binocular eyepiece. Many microscopes, in fact, we have some of these old ones in the lab that just have one eyepiece. So you only look through with one eye, which is more difficult. You're supposed to keep both eyes open. And when you're looking through with one eye, that other eye's looking, down at the lab bench. So that's not great. Our newer scopes, which are beautiful, again, you're going to have both eyes open, and you can actually adjust the distance between those, which is really nice, so that you're looking with both eyes at the object. So you're looking through, again, this piece here at the top, which you learned about back in week 2 in lab. This is called the eyepiece, and inside of here there is a lens called the ocular lens. If we move down our microscope, there is a second set of lenses here, okay, before the specimen, and those are called the objective lenses. And then the specimen sits right here on a slide on the stage. So the term compound in compound light microscope tells you that there are two lenses, compound meaning two, the ocular lens and the objective lens between your eye and whatever it is that you're looking at on that slide, the specimen. Okay? Let's talk about these objective lenses and the ocular lens. So first off, the ocular lens has a magnification power of 10x. It's going to take whatever you're looking at and it's going to make it appear 10 times larger, okay, than it really is. That's a constant. We can't change that lens. So the ocular lens is always 10x. Now the objective lenses down here, they're on a revolving nose piece. In fact, here's another one right here. So you can actually turn this, and when you turn it, you will actually put a different lens into place. And so the lenses there on the light microscope are going to have magnification powers of either 4x, which is the lowest magnification power. It's also called the scanning lens. 10x, okay, which is the next magnification up, and 40x, which would have been the highest magnification that we would have used in lab. And there is a fourth lens on there called the 100x objective lens. That lens requires oil. It's the oil immersion lens, so we tend to use that in the lab too much. 40x is really enough for what we would be looking at in the lab. Okay, so you can calculate the total magnification that you're using on the microscope, and you did this back in week 2 in lab, by taking the magnification of the ocular lens, which is always 10x, and here's the formula right here, and magnifying that by whatever objective lens is in place. So for example, if you're using the 10x lens, you would do 10 times 10. You would tell me that the magnification is 100x. If you're using the 40x lens, high dry, you would take 40 times 10, which is 400, and you would tell me that the magnification is 400x. Okay, total magnification. So it's those two lenses that work together to magnify the specimen. Okay, it's called a light microscope because at the very base here, there's actually a light bulb. So we use light here to illuminate our object from the bottom. Here's the specimen up here, so light's going to come up. There is another lens on our microscope called the condenser lens, which sits right here. And what that does is it takes and it condenses or focuses the light that's coming from the light source and projects it onto that specimen. Then that light passes through the specimen, and it's going to be collected through the objective lens. And light gets bent as it moves through these lenses, eventually making its way up to your eye. Okay? The benefit of using basic light microscopy is that you can view living organisms. You can view dead stuff too. You can view stained stuff. But again, the benefit, what you can't do with other types of microscopy that we're going to learn about, is view living organisms. So I can take, some pond water and throw it onto a microscope slide and put it on a light microscope, and I'll see organisms, mainly protists, right, moving around on that slide. I can view living organisms. I can watch organisms, in real time, which is pretty neat. Okay, this instrument operates on its ability to magnify, which again means to make an image larger, and its ability to resolve, which means to be able to see the fine detail of the object or the specimen as you increase magnification power. There's a limit here to the light microscope. It's not going to magnify over 1000x, right? So if I use the oil lens, the 100x lens, and multiply that by my ocular lens, it can go up to 1000x. But that's it. That's the limit of magnification. Now, let's say I want to see ultrastructurally. I don't want to just see the nucleus. I want to see the details of the nucleus. I want to see some of the proteins that are organizing the DNA. I want to see the actual ribosomes. I want to see ultrastructure, not just the outline and, some of the cytoplasmic material. I want to really see the details of that material. I can't use a light microscope. I've got to use something a little bit more powerful, and that's where the electron microscope comes into play. Now, the electron microscope, instead of using light, it's going to use electrons, negatively charged particles. Those negatively charged particles will be put onto the specimen or passed through the specimen, and you'll end up with a pattern that comes out of that, which again is called a micrograph. It would be an electron micrograph. With an electron microscope, you get higher magnification, higher resolution, and you could see more detail. The problem with electron microscopy is that you cannot view living specimens. Specimens are going to be killed in the preparation process, and the preparation process is difficult. Electron microscopy is costly. It takes a lot more money to maintain an electron microscope than it does to maintain a plain old regular light microscope. And we have our light microscope serviced once a year. They're not that expensive in the grand scheme of things. That maintenance is not too expensive. However, if you have an electron microscope, which we do not have at MVCC, again, they're much more expensive to purchase and as well as to maintain, and specimen preparation is much more costly and difficult. So we don't have one at MVCC. However, if you are a biology major when you get to your transfer institution, they may have one there, and they will have one, right? We have a cabinet of 20 light microscopes. We can afford that. But if your institution, your transfer institution has an electron microscope, they probably only have one. There's two types of electron microscopes, two types of electron microscopy. There is scanning, also known as SEM or scanning electron microscopy, and there's TEM or transmission electron microscopy. We're going to use scanning to look at the surface and transmission to look at the inside of cells. Okay, so before we jump into looking at the two types, I just want to show you the difference in images. So in image A here on our left, this is taken with a light microscope. Okay, you can see cells, and you can see what's shown in this picture, which is a bacterial infiltration of tissues. Okay, so all those little purple dots that you see there, those are all bacterial cells that are infiltrating a tissue. Now, look at B. All those little purple dots that you saw in A, all the little tiny purple dots, this is what they really look like. This is the ultrastructure. This is a transmission electron micrograph, just showing you the outside, but this is a rod-shaped bacterial cell right here. And here's another one here. And here's another one here. And here's another one here. And what are they doing here? They're getting inside the tissue. They're actually squeezing their way in between the cells here of whatever organism they're infecting. You can see the difference, right? Look at the magnification is incredible. The resolution, the clarity, the crispness of the image, the details. Electron microscopy gives this to us. Okay, it's beautiful. Okay, so let's take a look at the instrumentation differences first. And I'm just going to move my camera down here. So all the way on our left, we have a basic light microscope. Again, we looked at this. So you would look from the top, or you'd have a camera attached here to the top. And at the very bottom here, you would have a light source. So we're using just light. Okay, we're passing that through our specimen. With an electron microscope, what we have here is, at the base here, a film or a screen. And that can actually be captured. This can all be hooked up to a computer where it's being captured. And if we look at the opposite side, the top here of the microscope, we have an electron gun. So this is where electrons actually get shot out of. The electron beams are going to pass through some lenses that are going to condense them. And then we have our specimen sitting right here on our glass slide. And then electrons that pass through the specimen will be collected down here on our film or our screen. Okay, any electron-dense material are going to deflect or absorb those electrons as they come through. Okay, and then here is a scanning electron microscope, which is pretty much the same thing. Again, you have an electron source. Those electrons are passed through a series of lenses, and then you end up with your specimen down here. And any detected electrons get captured here. And again, you'll end up with a picture or an image on your computer screen. So this is, we're looking at here, a paramecium, which is a single-celled eukaryotic organism that tends to live in pond water. If we took a sample from around here, we'd find some of these guys. In the first image, let me move my camera up. In the first image here, this is taken with a light microscope. This is actually dark field microscopy, but it's just with a basic microscope like the ones that we would have in our lab. And you can see some pretty cool detail. You can see the plasma membrane, right? You can see the overall shape of the cell is pretty oval. You can see some stuff here in the cytoplasm. You might be able to see some organelles. These things right here, these are neat. There's two of them. They kind of look like the sun, right? Got a circle in the middle with some rays kind of going out. Those are called contractile vacuoles. And we're going to actually talk about those. Their job in a paramecium like this is to pump out excess water. So a paramecium living in pond water is going to have a tendency to take on water. We're going to learn in the next chapter, in chapter 5, about tonicity. The paramecium in this case would have a higher concentration of solutes inside of its cell compared to the water outside. And so water is going to move into that cell naturally because of the laws of diffusion. And that paramecium could swell over time from taking on water and then burst and die. But it's evolved a mechanism to take care of that extra water. And that's what those structures that look like suns, that's what they're doing. They're actually taking any of the excess water, instead of the paramecium kind of filling up, they're taking that water and they're just pushing it right back out. So they're pumps, they're water pumps, which is pretty neat. Okay, so you can see some pretty good detail there with a light microscope, good enough for us, right, for our level. But let's say you wanted to learn more about a certain structure, its shape, its location. Okay, an electron microscope comes in handy. So in transmission electron microscope, we want to see inside at those structures. So we would actually take our sample, our paramecium sample, and we would embed it in plastic, which is pretty difficult to do. And then we would take a knife A diamond knife would be the best knife that we could use, or you could also use a glass knife, but a diamond knife is the best. And you're going to cut ultra thin sections of your plastic block to cut through that paramecium to get slices of the cell. And then you're going to take those slices and you're going to hit them with heavy metal labels. You're trying to label certain parts of the cell. And so you're going to hit those parts of the cell with heavy metals and wash off any excess. And so anything here that shows up on this film is something that you labeled for. Okay? Electron dense material, that's those heavy metals. And so what you're seeing here, instead of just seeing in the cytoplasm, you're seeing actual structures. And some of these things might be mitochondria. We might have some lysosomes here, all kinds of stuff hanging out in the cytoplasm. So now we're not just seeing kind of fuzzy granular stuff, we're seeing actual structures. Okay, so in a transmission electron micrograph, we are seeing inside of a cell, at the structures inside of there. Let's say we just want to look at the surface though. Look at this. This is beautiful, this image. We're going to use a scanning electron microscope. And this is a scanning electron micrograph. Here we take the paramecium. We don't have to slice it in any way, so it's a bit of an easier process. Instead, we're going to coat the surface with heavy metals. And here we're staining the plasma membrane, and we're staining those little hair-like structures called cilia. We throw it under our microscope, we hit it with an electron beam, and this is the image that we end up with. Look at that ultrastructure. We're not just seeing the surface of the paramecium, we're seeing the actual cilia that are coming out of that plasma membrane. And right here, this little opening here is probably the opening to the food gullet, which is where food is going to enter. It's like the mouth of the paramecium. So you can see light microscopy is okay, but if you want to see ultrastructure either on the inside or on the surface of the organism, you're going to have to use electron microscopy. Again, the downfall of electron microscopy is that you've got to coat the specimen in electron dense material or slice the specimen up and then stain using electron dense material, heavy metals. And so therefore, you cannot use living organisms when you're dealing with electron microscopy. So if you want to see something alive, you've got to use light microscopy. Okay. Now microscopy is great. It tells us where things are in a cell, or it tells us what the surface of an organism looks like. But it doesn't tell us much about the biochemistry, or what's happening, what reactions are taking place, in which locations. Okay? It doesn't tell us what certain structures are made of, what types of enzymes are found in the nucleus, or what types of enzymes are found in the mitochondria. Okay? And so therefore, we've got to have a way to separate out cellular components, to study the nucleus by itself, to study the mitochondria by itself. That way we could learn more about what's happening inside of those structures. That's where cellular fractionation comes into play, and I just want to introduce you to this. Cell fractionation is a process in which we take a whole cell, or a collection of whole cells really, and we break those cells open and we separate out the component parts. And then those component parts can then further be studied biochemically. Okay, so if we want to know more about where something is or what something looks like, if we want to know maybe what it's composed of or what's happening inside of a structure biochemically, we would need to do this first. Okay, so cellular fractionation is pretty easy. What we do is we start with a test tube, and I'll start down here with a test tube. And what we do is we put cells into the test tube, and then we add some type of a buffer, usually a detergent. Soap is a great one. We'll add it right to the test tube, and we allow that to sit for a little while. And what's going to happen when we introduce a detergent, a buffered detergent solution, is we're going to break open the plasma membrane, and what's going to happen is the cellular contents, the cytoplasm and all the parts and pieces of the cytoplasm, right, all the organelles, are going to be dumped out. So we're just breaking the cells open, cellular lysis, okay? Obviously cells die during this process. Okay, so once we break the cells open and we have all the component parts now in this test tube, what we need to do is we need to spin the tube at different speeds for different lengths of time. And by doing that, we'll be able to separate out the component parts based on density. Okay, so cellular fractionation is based on what's called differential centrifugation, which is down here. Differential centrifugation. This is used again to fraction or separate out the parts of the cell. So we know that the nucleus, for example, is kind of the largest structure and the heaviest structure inside of the cell. So we'll take our tube full of all of our cellular contents, and we know that if we spin this tube at 600 Gs, that's the speed, for 10 minutes in a machine called a centrifuge, okay, then we know that we will pellet the nucleus. So we take our tube here. We usually have a bunch of these tubes. We put them into the hinged hanging bucket inside of a centrifuge machine. This is a large centrifuge. We turn the machine on and it's going to literally spin, okay, our tubes pretty fast, right? 600 Gs for 10 minutes. And at the very end, what we're left with is something that looks like this. Now, if we take a closer look down here at that tube, at the very bottom, there's going to be a dark structure called the pellet. And it's going to be condensed and kind of solid at the bottom of the tube. We know that our nucleus is down here, or our nuclei, I should say, because there's probably multiple cells in there. Again, this is called the pellet. It's a dense region down here. And everything above it is where all the other stuff in the cell is going to be. And that's going to be called the supernatant. Okay? So what we do next is we can take that supernatant off and put it into a fresh tube. And now we could just study the nucleus. Okay? Let's say we didn't want the nucleus. Let's say we want the mitochondria. Well, then we would take the supernatant in our fresh tube and we would spin again in our centrifuge. We would spin now at a faster speed, so 20,000 Gs for longer, 30 minutes. And again, the same thing. We would end up with a pellet and a supernatant. The pellet here would contain things like mitochondria, chloroplasts if we're dealing with plants. And the supernatant we can just siphon off and we can just study that pellet. Okay, we can go further and further. We can isolate each component part of the cell in this fashion. When we get to membranous parts of the cell, so the endoplasmic reticulum, the Golgi, which are structures that we're going to talk about, the plasma membrane, microsomal pellet, we can separate out these pieces as well using something called a sucrose density gradient. Okay, these sections of the cell will separate out based on density. We would spin this tube, and again, the plasma membrane would separate out towards the top of this density gradient. The Golgi would be towards the middle, and the ER towards the bottom. Okay, of course, we have to spin this one more time. So again, you would use cell fractionation or differential centrifugation to separate out component parts of a cell so that you can study them further biochemically to learn more about what's happening inside of them or what they're made of. Okay, again, the machine used here is called a centrifuge. It's a machine that spins samples at various speeds for various lengths of time. Okay, so let's move on to the cell theory. Okay, we know what a cell is. We know that we can look at cells. We know that we can separate out cellular components to study them biochemically. So the cell theory. Back in 1665, a scientist by the name of Robert Hooke, he was the first scientist to see non-living cells, unfortunately. He was looking at cells of cork, which are dead plant cells. And when he looked at these cells, he noticed the rectangular, really rigid box-shaped structure of those cells. Again, they were plant cells, so they had cell walls. And so he thought, this is a terrible story, he thought that those cells, right, that we call them now cells, that they looked like the rooms in which monks lived in. And so that's where the cell got its name from. The rooms that monks lived in were called cells. Okay, and so that's where it got the name from. Again, terrible story, but that's how they were named. There was another scientist who gets credit for being the father of microscopy for actually viewing the first cell. His name was Anton van Leeuwenhoek. He saw the first living cells, but again, Robert Hooke saw the first dead cells. Okay, in the 1830s, we have 3 more scientists that come into play: Schleiden, Schwann, and Virchow, and they're responsible for coming up with the cell theory. Robert Hooke named cells, but Schleiden, Schwann, and Virchow came up with the cell theory. The cell theory has really 3 parts. First off, the cell theory states that all living things are composed of cells, from the simplest single-celled bacteria to the most complex forms of life, like humans. We are composed of cells. Second, the cell is the basic unit of life, which means that cellular components cannot live on their own, but cells can. Okay? Single-celled organisms, that's the smallest form of life. Again, a nucleus on its own is not considered to be living. A mitochondria, or any cellular part on its own, is not considered to be living, but cells are. And lastly, new cells come from pre-existing cells. There is no such thing as spontaneous generation. Cells don't just poof, arise out of thin air. Instead, they come from pre-existing cells. So that would be through mitosis or meiosis, which we're going to learn about in a few chapters, or through sexual or asexual reproduction. Okay? That is the only way that new cells can come to be. So let's talk about prokaryotic cells first, and then we'll talk about eukaryotic cells. So prokaryotic. Pro means before, and kary means nucleus. So prokaryotic cells probably arose first. Okay, that's what evolutionary theory tells us. And that these cells, which were probably some of the first cells around on the planet, that they did not contain a membrane-bound nucleus, nor did they contain any other membrane-bound compartments. So when we talk about a nucleus or mitochondria, chloroplasts, or the Golgi or the ER, which are all structures that we'll talk about a little bit later in this chapter, these are not found in prokaryotes. Okay? The domains archaea, which you've heard now, and bacteria, remember the archaea were single-celled prokaryotic organisms that are the extremophiles, right, that like to live in extreme environments like hot sulfur vents, or extreme salt environments, or environments that are really high in methane, for example. Those are going to be single-celled prokaryotic organisms belonging to the domain archaea. And then you've all heard of the domain bacteria. You've heard of bacteria in general. Those are also going to be prokaryotic cells. They are all single-celled organisms that lack organelles. They lack a membrane-bound nucleus, namely, and they also lack other membrane-bound structures. Okay? So that's the key difference between prokaryotic and eukaryotic cells, is the presence in eukaryotes of membrane-bound structures. However, it's really important to stress the similarities or commonalities of all cells. So whether it's a prokaryotic organism with a prokaryotic cell or a eukaryotic organism with eukaryotic cells, there are going to be a handful of similarities between them. And this is a really good indication of evolution. In fact, there's a lot of similarities between prokaryotic and eukaryotic organisms, not just in structural entities, but also in biochemistry and in metabolism, reactions that take place. A lot of similarities. Okay, so here's a couple of similarities if we're looking at just cells or cell structure. So first off, all cells have a plasma membrane. And we're going to dive deep into the plasma membrane in Chapter 5. We'll talk about it here a little bit. But the plasma membrane, whether it's a bacteria, a prokaryote, or a eukaryotic organism like us, all of our cells have a plasma membrane. And that plasma membrane is going to be made up of a phospholipid bilayer. You saw this back in chapter 3. A phospholipid bilayer that has embedded in it proteins, carbohydrates, sometimes molecules like cholesterol. Okay, so again, all cells, prokaryotic and eukaryotic, contain a plasma membrane that is going to surround the internal contents of the cell. It's going to be that barrier between the cytoplasm and the outside environment. Now, some cells, plant cells, and some prokaryotes are going to have structures outside of that plasma membrane, a cell wall or some other type of a structure, a capsule, something like that. But again, all cells will have at least that plasma membrane. The second thing is all cells have a cytoplasm. All cells have a fluid structure inside, to the inside of that plasma membrane where reactions take place and macromolecules are found. Okay? Third, and this is probably the most important on this list because sometimes students will get kind of hung up here, all cells, prokaryotic and eukaryotic, contain DNA. All cells contain genetic information. Now, the structure of that genetic information is different. So in prokaryotes tend to have one circular double-stranded piece of DNA or chromosome, where eukaryotic organisms tend to have multiple double-stranded linear chromosomes. So again, prokaryotes, you've got one circular double-stranded piece of DNA. In prokaryotes, you tend to have more than one linear double-stranded DNA or chromosomes. Okay, so there is a difference there, but what is shared is that all organisms have DNA, and that DNA is always double-stranded. Okay, and the last thing on this list is ribosomes. So all cells that are living have these structures called ribosomes. And we're going to look a little deeper at ribosomes in this chapter, but just to give you a heads up, a ribosome is going to be a structure in which proteins are going to be synthesized or produced or made on. Okay, they're considered to be protein manufacturing machines. Okay, so there's the commonalities. Let's take a look at a prokaryotic cell. This is a typical bacterial cell here in the picture. So again, prokaryotes, they're going to be simple. They're going to be unicellular. Okay? They don't have a membrane-bound nucleus. However, they do have DNA, and that's shown here in this picture. This is chromosomal DNA, and you can't see it. It's one big circular double-stranded piece of DNA. And that DNA tends to be found kind of centralized or localized in a region of the cell called the nucleoid. Now, there's no membrane surrounding that, but it is usually contained to a region. Okay? Different from eukaryotic organisms like us, our DNA is enclosed in a membrane-bound compartment inside the cell. And that's fantastic because our DNA has a higher level of protection, and we can really compartmentalize DNA replication and other processes like transcription. But bacteria do pretty fine without that. Okay, so they have DNA, it's double-stranded. It looks a little different from eukaryotes, and it's definitely not in a membrane-bound region of the cell. They don't have any other membrane-bound organelles, so you're not going to find mitochondria. We're going to learn that the mitochondria is the site of cellular respiration in eukaryotes like us. Prokaryotes still do respiration. It's a little different. They don't have mitochondria, and they tend to do that respiration on their plasma membrane. That's usually the location of it. Okay, so processes are still similar, but they're not the same. They're not going to happen in the same locations because of the lack of those membrane-bound organelles. You're not going to find structures like lysosomes or endoplasmic reticulum or the Golgi body in prokaryotes. So that's what makes them simple. They have less compartmentalized spaces inside of their cell. Okay. Okay, so some or most prokaryotes have some other stuff. Outside of their plasma membrane, many of them will have a cell wall. The cell wall is going to look different than the cell wall of plants. The cell wall is going to be composed of a structure called peptidoglycan. Okay, again, a little different than the cell wall of plants. The point or the purpose of the cell wall here is for protection. It helps to maintain cellular shape, and it also prevents desiccation or dehydration, or it prevents the cell from drying out. Okay, so it helps to prevent water loss. Some bacteria outside of that cell wall will also have a structure called a capsule, and that capsule is going to be made up of a carbohydrate. Okay, so polysaccharides. The purpose of that capsule is to aid in attachment to surfaces. Okay. Some bacteria will also have a flagellum, or sometimes two, and that's this structure here. It looks like a whip-like tail, or flagella, if there's multiple, are going to aid in movement. And that movement might be pushing an organism or pulling. Okay. What else? Some will have these little structures here. They look like cilia, but they're not. They're called pili. And these pili are going to be involved in the exchange of genetic material, usually these little tiny round circular small pieces of DNA called plasmids. Sometimes those get exchanged. They get moved from one bacteria to another. Those pili are going to be what are doing the exchange. So it's going to be a connection made between two neighboring bacterial cells, and that is called conjugation. Okay, we don't call that sexual reproduction, because it's not sexual reproduction. It's just the movement of genetic material from one bacterial cell to another. Okay, and again, this usually happens on small, mobile DNA called plasmids. Okay, and lastly, some bacteria will have other structures, which I don't think are pictured here, called fimbriae. And those fimbriae are going to be used to attach to other cells. Okay, so the capsule will help the organism to attach to surfaces. Fimbriae helps attachment to other bacterial cells, or other cells in general. Okay, so again, pretty simple. Much less complex than eukaryotic cells. But again, I told you that all cells are microscopic, right? Mostly all cells are microscopic. So cells on average are between 0.1 and 0.5 micrometers in diameter, which again, you can't really see with your eyes. You need a microscope. Okay, let's take a look here. So you, or I, adult female here, we can see, right, adult humans with our unaided eye, we are that size. You could also see cells like an ostrich egg and a chicken egg and a frog egg. And we could put over here maybe even some nerve cells, large nerve cells. Those are cells that we can see. So the egg of many organisms, even the human egg, it's going to look really, really small. You can't see any details, but you can see it with the unaided or naked eye. Okay? But most cells, like plant cells, animal cells, you can't. You need a microscope to see those. Okay? A light microscope is pretty good, but if you want to see ultrastructure, if you want to actually see the phospholipids of the phospholipid membrane, or you want to see the proteins that are in that membrane, you got to go down and you've got to use the electron microscope. Much higher resolution and magnification. Okay? Bacteria you can see are smaller than most plant and animal cells. Bacteria on average are about the size of our mitochondria. Okay, and if we go down here, the flu virus, interesting. So the flu virus or the coronavirus, right, we would need an electron microscope to see those. And these viruses we consider to be non-living. They don't possess all the characteristics that living organisms possess. Okay, So prokaryotes as well as eukaryotes, our cells usually are going to be microscopic. We are going to need some type of microscope, either a light microscope or an electron microscope, to really see these cells. And the question is why? So why do our cells remain so small? And that's because we have to have a high surface to volume ratio, so enough membrane to service the cytoplasm of our cells. We need to be able to bring in nutrients and materials and get rid of waste products, so excrete waste products from our cells quickly in an efficient manner. If our cells grow too large, there won't be enough surface area, enough membrane, to be able to handle the demands of the cell. So we want to keep a high surface to volume ratio. We want to have a lot of plasma membrane present to bring stuff in and to get rid of stuff so that the cytoplasm of our cells can remain efficient. Okay, and that's why our cells haven't gotten so big. Here's a nice diagram just to show you this so that you can actually see what surface area to volume ratio looks like. So let's say we had a cell that was this size, and then let's say we took that cell and we broke it up into multiple cells, right? We have the same area here, okay? Different number of cells. So if we look at surface area to volume, okay, we just take a look down here at the bottom here. I'm not going to ask you to calculate these. But if we just look at the overall ratio here at the bottom, the surface area to volume ratio for this one larger cell is 3. Okay, whereas our surface area to volume ratio for all of these is 6. So if we take each one individually, and we look at the surface area to volume, we end up with 6, a higher surface area to volume ratio. So these cells on this side are going to be more efficient at bringing in nutrient materials and at getting rid of waste products. This plasma membrane here only has to service that space of cytoplasm, where this cell has a lot more cytoplasm to service. Again, this is all based on surface area to volume ratio. The higher that number is, the higher the surface area to volume ratio is, the more efficient that cell will be. Okay, so staying smaller in size definitely has an advantage. Okay, so we're at our first breaking point. So again, I want you to pause the video here. You've got a series of questions to answer on this Are You With Me? And when you're finished, you're going to come back and I've got some questions for you. Okay, so here we go. Here's some questions. First one, when viewing a specimen through a light microscope, scientists use blank to distinguish the individual components of cells. Do they use a beam of electrons, radioactive isotopes, special stains, or high temperatures? So think about this. I don't know if I mentioned this too clearly. I know that I told you that with a light microscope you could see living organisms, which is pretty neat. But if you want to see structures, and take us back to week 2 in lab, we did some of this, or I did some of this for us. We can stain for certain structures. And sometimes those stains don't even kill the cell, so you can keep a cell alive while still staining. The one that we used, or I used, in lab week 2 was methylene blue on my cheek cells. And that's going to be used to stain the nucleus of eukaryotic cells. So pretty neat. So for this question, if you're using a light microscope, you wouldn't use a beam of electrons, right? That's electron microscopy. We never talked about radioactive isotopes or high temperatures. So special stains are sometimes used in light microscopy to see different structures. Okay, second question. The blank is the basic unit of life. The organism, the cell, the tissue, or the organ. Hopefully you answered blue, the cell. So the cell is the basic unit of life. Got a couple more questions. Second question. You want to determine the location of a specific protein in a living cell. There's your clue word, a living cell, using a colored stain. Which of the following is the best technique for this? Would it be TEM, transmission electron microscopy, SEM, scanning electron microscopy, light microscopy, or density gradient isolation? So that term living gives you your clue here. So hopefully you chose green, light microscopy. Next question. What is one cell structure found both in prokaryotic and eukaryotic cells? Is it the nucleus, chloroplast, mitochondria, or the plasma membrane? So hopefully you answered yellow, the plasma membrane, found in all cells. Next question is about cell size. So which statement below about these figures is true? Okay, does figure B have a smaller volume than figure A? Okay, or is the volume the same? So that collection of cells in A compared to that one cell in B. Okay, does B have a smaller volume than A? Does figure B have a greater volume than figure A? Green. Does figure B have a greater surface area to volume ratio than figure A? Or does figure B have a smaller surface area to volume than figure A? So think about that for a minute. Remember back to that picture that we looked at. So if we look at A, you've got a bunch of cells there, but is the surface area to volume ratio, is it larger or smaller than B? Okay, or if you look at A, is the volume greater or smaller than B? Which one of these is correct? You may have to read them again. You can pause me if you need to. The answer here is yellow. Figure B has a smaller surface area to volume ratio than figure A, or figure A has a larger surface area to volume ratio than B. Okay, you've got much more surface area in A compared to B, but the volume is the same. So that collection of cells in A has the same volume as the cell in B, but look how much more surface area there is, okay, compared to that volume. So the surface area to volume ratio is greater in figure A, that collection of cells, when compared to figure B, that one single cell. So again, yellow would be correct. And just to hit this home, the reason why that is important is because the greater the surface area to volume ratio is, the better those cells will be, again, at servicing the cytoplasm of the cell. So the better they'll be at bringing materials in, raw materials that are required for cellular maintenance, cellular homeostasis. Having a greater surface area to volume ratio also will help to get rid of waste products more efficiently. Okay, so cells will run more efficiently in that case. Okay, so moving on, and actually I think I'm going to save this for my next video. So I'm going to go ahead and stop here, and I'll start the next video talking about eukaryotic cells.